Good morning, everybody. How's it going? Let's see here. Who do we? How many? We have 25 students in class. Good morning. Let's see here. Just want to take a peek. The syllabus. Yes. So today's the 18th. And yes. Okay. Sorry, I just was getting situated with the syllabus. I wanted to see how we're doing. Because honestly, we're doing good. And I'm thinking, okay, all right. Let me get this away. All right. Good morning. Wow, everyone, so many people said good morning today. Good morning. Let's see. Part 1. Sorry guys, just give me one second. I just want to see one thing. One second. Let's see something here. So we have today's class, and then 1, 2. Okay. Yeah, all right. Let's get started. Sorry about that. I just wanted to see where we were, so that I could see if what I had to get through today for us to be doing all good. Oops, wow. All right, let me fix this thing. Great. This in the corner. So yeah, there was an assignment due. There was an assignment due this morning by 10 a.m. If you missed that deadline, just make sure you email me right away so that I can reopen it for you. Yeah, so make sure you email me as soon as you can. Last time we did the Horsetails, which are super exciting. And we talked about the steel again, which is super exciting. And then alternation of generations in the Horsetails, right? Today we're going to talk a little bit about the ferns. I'm not sure if we're going to get through all of this today, but we'll at least get it started a little bit. So let me just make sure. Yeah, good. I always like to get oriented, right? So we have worked our way where we've left the non-vascular plants, are now back way in the past. We're in the vascular plants. We're in the spore shedding vascular plants, and we've already done the lycopods, we've done the horsetails, and now today we'll start talking about the ferns. The ferns are in the phylum Polypodiophyta. That's how you say that. And they are one of my favorite plants, probably, if not my favorite. I almost would have just studied ferns only because they're just so cool. The name Polypodiophyta comes from the words poly, which means many, and then podi, which means foot. So it's the many-footed plants. And the name also is the same as large genus called Polypodium, which is one of the largest genera, one of the largest genera in the ferns. And there's lots and lots of species, about 11,000, and there's a ton of variation in terms of things like size and habit or shape and habitat, which is where it lives, like where it lives at. Okay, so here's size. So look, you can have, these are both examples of ferns. On the left, you see Azolla, and on the right, you see, okay. All right, on the right, so on the left, you see Azolla, and on the right, you see Salvinia, but both of these are plants. These are full-grown, mature plants, and they're on fingertips here, on somebody's fingertips here. So some ferns can be super, super small, like a few millimeters to a few centimeters in size, such as these two. But then you have some ferns that can be the size of a tree, right? So like here, we see these tree ferns along the river of here, like those giant trees are each ferns, and they can get up to like 60 feet tall. So a lot of diversity in size. Also a lot of diversity in form, or what we can say habit. Habit means form. So some fern species are epiphytic, meaning they grow as epiphytes on other trees and other plants. We've come across that term a lot before. Some ferns are aquatic in that they grow in water, either submerged in the water or emergent or coming out or even floating on the water. Some have a form which is being like a small herb. This is probably the most familiar form of a fern, right? When you think of a fern, maybe you think of something you see as you're walking through the forest or something like that, and it's like this little tiny herbaceous plant, right? That's what you might think of. This is my common form. Tree-like forms as well, as I mentioned already. And then now, so that was habit or form, and now we'll talk a little bit about habitat. Habitat is where the things live, where something lives. So for ferns, some of them have a really tropical habitat, so like a rainforest or something like that. You can see here on the bottom is just showing you that there are certain species of animals that actually really depend on some fern species, like some species of frog live almost their entire life cycle inside of ferns. And there's some big giant ferns that make kind of almost a nest shape, and some species of bird actually build their nest on top of these ferns that grow as epiphytes in these tropical habitats. And then of course in our region, both New Jersey, New York region, and then out where I am in Michigan, they grow as temperate habitats. So forests, temperate forests, very common understory plant, something that you see quite a bit. And look at this little cute deer is actually eating fern right here, eating some ferns. All right, so some familiar things here is that we know we're in the vascular plants, so we know that the Sporophyte generation is dominant and long-lived. We know that it is vascular, and we know that it has true roots, stems, and leaves. We already know all that. We know we're in the spore-shedding vascular plant, so reproduction is via Sporangia that shed spores that grow into gametophyte plants, right? Most species are homosporous and exosporic. Some species are heterosporous and endosporic. Usually the aquatic species are heterosporous and endosporic. So just a little bit of a reminder, remember whenever you see these terms, so homosporous and heterosporous refer to the sporophyte plant. Refer to the sporophyte plant because the sporophyte plant is what is shedding spores. So if a sporophyte plant is homosporous, that sporophyte plant is going to shed one type of spore. If the sporophyte plant is heterosporous, it's going to shed two types of spores, right? Whereas things like exosporic, and endosporic refer to the gametophyte plant, because the gametophyte plant grows, in the case of exosporic, outside of the spore, and in the case of endosporic, inside of the spore. So just a little bit of a reminder there. But if a species has homosporous sporophyte plants, it's going to have exosporic gametophyte plants, right? So that's why we say here with the homosporous / exosporic. Okay. So yeah, growth by apical merosomatic cells. Okay. Yes, ferns. So some of you may even have a fern. Maybe you even have a fern in your house. They're very, very common house plant. And so we actually have a lot of, they're economically important that way, because we do use them in both house plants and in landscape plants as well. Remember the name, the phylum is Polypodiophyta. Polypodiophyta. Part of that comes from the way that the rhizomes look in some species. So remember, a rhizome is a horizontally growing stem, right? A stem that grows horizontally. And in some fern species, so in the fern, sometimes the rhizome will be above the ground that you can see with your eyes, and other times it will grow below the ground. But in any case, a lot of fern species rhizomes, so like here's one rhizome that I just circled, it's like right here, right? Here's another one right here. The rhizomes actually kind of look like, almost like fuzzy feet. Like you could think of like a rabbit's foot or something like that, little fuzzy feet. So like here, this is one. And look how many are on this one fern. So I think part of this is why it has the phylum name Polypodiophyta, because it looks like lots and lots of little like animals' feet. Got a question in the chat box. Looks like a hairy spider. Yeah, I know. It does. I actually have I have one type of fern. Maybe next time I'll try to like see if I can grab it and put it for us to like look at, because it has this same look of over here. Oh, I gotta get rid of this chat box. It looks like this on the bottom of the fern, but it's not fuzzy so much as it's like, it really does look more like less hairy spider. But yeah, totally. Yeah, so I think that's where this idea of like polypode, like many foot, showed up, because of the way the rhizomes look. So more diversity. So ferns are incredibly diverse. It's part of the reason why I find them so captivating, because they got a lot going on for them. If you study ferns, you're never going to be bored. There's like so much diversity. And here's another example of that. So ferns have megaphylls, which we will talk about in a little bit. And look at the diversity. So this is just an example of some forms of megaphylls, so like what they look like. And it looks in so many different forms. Really interesting and pretty. So yes, this new term. So this is a new term for us here, megaphyll. Megaphyll. Let me change colors because I got this weird green background on this slideshow. Megaphyll is a new term for us, megaphyll. It might look familiar because it has components that should seem familiar already, but the term itself is new for us, and we'll talk about why right now. So remember back lecture 4 when we were, I think it was lecture 4, maybe it was lecture 5. It was the lecture where it was the introduction to the vascular plants. Here, I'll tell you right now, actually, just so I can make sure and not lead you astray. I think it was lecture Yes, it would be lecture 5. That was the introduction to the vascular plants, the plant body. Yes, so I just looked at the syllabus here. Yes, remember back in that lecture where we talked about the microphyll, right? The microphyll, and how we think based on the fossil record that the microphyll came to be. And so what we think is that there were these things called emations, which we see in the fossil record, which are these kind of pokey things, right? Here's a picture we saw before from an extinct plant with emations, and all of these pokey things coming off almost look like, oh, I should do red. Almost look like little thorns or something like that, right? Here's one, here's one, here's one, right? Emations. And so what we think happened is over evolutionary time, here we go, evolutionary time, we see this transformation in that the vascular tissue, which in this drawing, just as a refresh, this darker area in the middle is the vascular tissue, this stuff here. And in the enation, it's not present, right? But then ultimately it kind of starts to migrate in. And once the vascular tissue migrates into the enation, no longer an enation anymore, it's what we call microphyll, which we're used to because we've come across microphyll a lot. So microphyll, remember, it's that leaf that has a single vein, very simple, right? This is what we think happened in terms of evolutionary history of a microphyll. But now, as we saw on the previous slide, now we have a new term, megaphyll. Or depending on what book you use, you might see macrophyll. For our class, we say megaphyll. So unlike a microphyll, a megaphyll has many veins, not just one, many veins. And it's, you might have guessed it, larger than a microphyll. And megaphylls and microphylls are two separate things. And megaphylls did not evolve from microphylls. They have two separate evolutionary histories. Megaphylls came about via the fusion of branching systems. So microphyll came about via vasculature moving into an enation. Megaphylls, it came about via a fusion of branching systems. And that fusion involved 3 steps: overtopping, planation, and webbing. And I'll show you what those 3 things mean. So before that, let's recall what these really earliest vascular plants looked like, right? And they had this equal dichotomy. They did not have any leaves, right? They had photosynthetic stems. And hold on, that's my husband making noise. So right, that's what the earliest vascular plants looked like, right? And we got to get oriented here. Bye. Let's see. Leafless, and yes, so photosynthetic stems, leafless, etc. However, eventually you would have had some branches kind of growing taller than the other ones, right? As you can see in this next drawing. And those more aggressive branches would overtop the weaker branches, right? So that's what we say, overtopping. And you can see here, who here is overtopping? Who here is overtopping? This one, right? This one, that's not a great circle. This one here is overtopping, right? Because it's higher than these, and it's higher than these, right? It's overtopping them. It's higher than them. So that's the first step in the evolution of a megaphyll, is this overtopping. Next, you have this kind of flattening occur. So I'm going to try to do it, and here, maybe I'll stop share for a second so you can see my hands. But like, okay, so like overtopping, so let's say here's your branches, right, like this. And in overtopping, like this one grew faster, and so it's higher up than this one. And then planation is flattening. So here their branches are kind of like three-dimensional. In planation, it kind of just flattens out like this, right? Like this. That's planation. So like three-dimensional structure to kind of a flattened two-dimensional structure. That is planation. And that's what this, oops, that's what this is showing you. So when I put my hands out to make them flat, that's kind of what this here is trying to show you. These branches just kind of flattening out. Flattening out. And that's the second step of the evolution of megaphyll. Last but not least, the flattened branches would have had some webbing occur, or they would have become fused together. And once that occurred, the result is, so here's that, oops, here's that webbing right in the middle, and the result is a megaphyll. It's a megaphyll. Whew. Oh, finally. Whew. Okay. Sorry about that. So megaphylls, so when something occurs in evolutionary time, it doesn't mean it just happened at one time and then everything else has it, right? So megaphylls, for example, evolved independently at least 3 times. So this process of overtopping, planation, and webbing occurred separately in the horsetails, in the ferns, and then separately in the seed plants as well. So it evolved independently at least 3 times. So here's what megaphylls, some examples of megaphylls. So you can see much bigger than a microphyll, much bigger than a microphyll, many veins. So like here, this is showing you one megaphyll. And look at on the inside, all of the veins, they're the lighter thing. I'm drawing on some right now, right? This here is a close-up of part of a megaphyll, because the rest of the megaphyll would keep going down here, and you know, these are all the different veins here. Yeah, but the point is much bigger than a microphyll and lots of veins, We're not going to go crazy on the steel right now, but I just want to show you guys, because you'll see it eventually in lab, as a recall here. So we know the steel is how you describe the organization of the vascular tissues in the stem, right, in the shoot system. On the left, you have protosteel. That's the most basic one, and that's what Lycopodium has. Next to that is siphonosteal with no leaf gaps. It's a little less basic. That's what Selaginella has. The time before this class, we had the horsetails, and we learned that Equisetum, which is the only genus in the horsetails, has a combination of this siphonosteal with no leaf gaps and a eusteel. And now we're in the ferns. And so most ferns have a siphonosteal with leaf gaps, which is this third one here, right? Siphonosteal with leaf gaps. And although there's some primitive ferns have kind of like more of an advanced protosteel, but most of them have this siphonosteal with leaf gaps. The reason why they have leaf gaps and as opposed to no leaf gaps is because ferns have megaphylls, and the others that we have looked at had microphylls. So yeah, we'll see what this means on the next page, in the next slide. So here you go. So let's say you did cross-section through Lycopodium, right? Shown here on the right. And you do this cross-section and you make a microscope slide. When you look at the slide, you'll see the vascular tissue here. I guess I'll use yellow, pink. Let's use pink. Right, so you do cross-section through this lycopod right through there. Here is a schematic of that. You look at it under the microscope, and here's what you see. You'll see the protosteel here, which is that solid, dense vascular tissue, completely solid. And then if there's a little microphyll, see this is a microphyll, and you happen to get part of that when you're doing the microscope slide, you'll see a little vasculature right there where the microphyll would have grown out, right? So like this part of the vasculature right through there, that's that little dot. However, if you were to do a cross section through a fern stem, so here we're showing you we're doing a cross section through the rhizome, which remember is that, look at these orange, horizontal stem. So look, we're doing cross section through the horizontal stem down here, which is the rhizome. And it's shown right here. But however, now ferns have a megaphyll. And so the vascular tissue is different. They also have a siphonostyl with leaf gaps. And remember, a siphonostyl has a ring, right? It's a ring and it has a hole in the middle. And so not solid. And if you were to look at that under the microscope slide, so like this part here, that little C is the stem vasculature. But then look, check this out. Look at this part here, right? Here, I should draw on the actual thing, not right. So you have this. It's a xylem and phloem inside of the stem. And then this littler thing here is your, is showing you where that megaphyll is coming out. And this hole in the middle is called your leaf gap. So hence the siphonostyl with leaf gaps. That's the leaf gap. I'll show you what it looks like even more. Oh yeah, so we call the hole is the leaf gap here. The hole, it'll be clearer on the subsequent slides, but like this hole right here, middle is the leaf gap, but that little part of the vasculature is the leaf trace. Here you go. So this is what it looks like in a real microscope slide. And you can see this siphonostyl is not in a full ring anymore, right? Siphonostyl is in this kind of C shape. And then there's this nothing, there's nothing right here. And that nothing is your leaf gap. Leaf gap is right here. And you can't really see the leaf trace on this microscope slide, but you can here. Here you go. So thing on the bottom, thing on the bottom is the stem vasculature. Thing on the top is that leaf trace. And the hole in between would be the leaf gap. So siphonostyl with leaf gap. And that's what that looks like. Take a sip. I'm gonna see how we're doing here. See how we're doing here. Okay. Yeah, all right, we're doing good. We're doing good. I think we'll probably finish a little early. So let's keep going. Oh wait, I stopped sharing. Can't see the screen. Share. Okay, good. Cool. Okay. So these are ferns, just to kind of like remind you what we're talking about in the first place, because it's all this siphonostyl stuff. Yes, pretty ferns. So, Sporophyte generation of the ferns, right? The shoot system, and is what we'll focus on first. And we just learned this thing, megaphyll, right? Megaphyll, aka leaves. So everything else that we're going to talk about has megaphylls anyways. Only Selaginella and Lycopodium have microphylls. So everything else that we say has megaphylls, aka a leaf. And the leaf has different, or AKA leaf, AKA frond. You might see frond in the lab stuff. And the leaf has 3 parts in the ferns, and it's blade or lamina, which I don't know if you can really see here, but it's the green part. So like the green part, the blade or lamina, the rachis, which is the stem inside of the blade or lamina, and then the petiole, which is the stalk. There'll be more pictures of this on later ones that could be easier to see. Okay, and there's a lot of variation in the leaf. Whoops. Let's see here. Oh, I had a question in chat box, but I think it went away.
Hi, Professor.
Hi.
I just asked if you said that only Lycopodium and Selaginella have microphylls.
Yep, that's right. That's right. They're the only ones left on Earth that have them.
Okay, so what about horsetails?
Right, right. Well, so remember, oh, good question. Here, I'm gonna pull it up real quick. That's because, yeah, so horsetails technically have megaphylls. Let me pull up this slide from last time. Here we go. Yes, okay. I think I might have to share this one separately. Let me see if I do. Yes, I do. All right, I'll share this one separately. Share. Can you see this slide showing the horsetail? It says leaves very small, etc., like that?
Yes.
All right, cool. So remember, so this is from the horsetails. We saw this last time, but it is kind of confusing, but so we'll look at it again right now. But look at this. So here, go away. Sorry, I'm everything. I need to have like a TA like manage the tech here, you know, like get my chat box out of the way, help me annotate. So we looked at this last time. This here is, these are the leaves of horsetails, right? Here's some more here. And yes, they look in appearance to be like a microphyll because they are small and they're simple and they have one vein, right? They're small, simple, and one vein. However, we know now based on the fossil record that the leaves that are on horsetails that are still alive today are what's left over of megaphylls that the relatives of horsetails had millions and millions and millions of years ago. So if I go back here, in evolutionary time, what happened was, I think yellow is probably good. This would have been one leaf, and all of these would have been its veins way back in the day, But because of different selection pressures, that having a big leaf like that didn't work out well for the horsetails. And so they eventually just they lost all the lamina and they basically have only the veins left, such that these are now what we call the leaves. But technically, their evolutionary history, so these did not, here's a better way to say that. The leaves and horsetails that we see with our eyes, yes, they're small, yes, they're simple, yes, they have one vein, but they never were an annation. They did not evolve from an annation. They evolved from the overtopping, planation, and webbing. So they are technically megaphylls. And if we stop sharing this and go back to here, where's my ferns? Go back to the ferns, which I think, am I sharing it? No, share. And make it go back to this here. Yep, so here you go. Yeah, so megaphylls evolved independently in the horsetails, and then in the ferns, and then in the seed plants. So good question, because it's good to clarify that. Yes, only only Lycopodium and Selaginella, aka the lycopods, have microphylls. And that's also why Lycopodium and Selaginella have these types of vasculature in the stems, because these things here are for microphylls. But once you have megaphylls, you'll have these leaf gaps. Does that explain things, Brittany? Did I confuse you even more?
Thank you. That was really helpful.
Okay, great. I'm happy to hear that. Sometimes I get carried away. Yeah, so it's good. Like I like this slide a lot, because when you see this slide, this figure comes from the book, and it's not very exciting. It's not a very exciting figure, and it has these words on like stephanosteel. It's just not super exciting. But then when you get here, this is great. This is really good because it really shows the difference of having a megaphyll versus a microphyll, and what that'll look like under the microscope. All right, leaves. Okay, we have a lot of diversity in the ferns, and I mentioned earlier that one of the ways is in leaf form or shape. Here is some examples. This is not all of the examples, but here are some examples just to get you started with having some of this terminology. So we're going to talk about different varieties of leaves, or variation on the frond, but varieties of different leaves that the ferns have and how we can describe them. So number one, here's a simple leaf, right? Again, these are all megaphylls. All the ones we're looking at now have multiple veins and they're megaphylls. So simple leaf. When you see simple leaf, you guys are gonna make fun of me, but like if you ever have played with a leaf and you hold the stem that the leaf is attached to, right? So like this thing here, you hold the stem and you're playing with the leaf. If there's only one thing above that stem, so here, this has one thing attached to the stem, then it's a simple leaf. However, some leaves have more than one thing. If you're holding the little stem, might have more than one thing, right? So like this has these things on the stem. This has all this on the stem. This has even more stuff. So if there's a lot of stuff above the little stem thing, here's a little stem thing down here, then we call it a compound leaf. There's different type, as you might look at these three examples here, these all have more than one thing above the little stem, but they otherwise look pretty different. So there's different ways to describe the different levels of complexity in the compound leaves. So here's one type. This is palmately compound. The reason we say that is because if you were to hold this leaf, it's kind of shaped like a palm, meaning like flat. Like a stick, and then everything comes out from the same spot, right? Meaning, look. Let's use bright blue, I guess. So like, see here, look, stick, and everything that comes off of that stick is coming from the same spot. This going this way, it's going that way, same spot. Kind of like a palm, right? Look, we had what? There, it's a hand. Palmately compound, that's one type. But the more common compound shape is something called a pinnately compound. Pinna means feather, and I think this is aptly named, right? These look like feathers. To me, they look like feathers. So you can have pinnately compound leaves. These terms are really useful if you're ever like, let's say you're walking around somewhere, anywhere, and you see a plant that you just think is so beautiful, and you really want to know the name of that plant, and because you want to grow it someday, right? And you're like, gosh, what is the name of this plant? Terms like this, being able to look at the plant and be like, well, it has simple leaves, or it has compound leaves, or it has a palmately compound. Being able to see that on a plant, well, if you search it on Google or use a plant book to identify it, really helps narrow down your searches. So I'm just letting you know that, as versus searching like plant with red flower, you know, just, yeah, good luck with that. So it's helpful to have these terms. Pinnately compound looks like a feather. Sometimes there's different levels, so for certain species. So this one is 1 times pinnately compound, in that here is the stem, and there's just 1 level of complexity, 1 level, right? Just 1. Meaning off the compoundness is just directly attached to that first stem, right? There's just 1 level, which will make more sense if you see this, which has 2 levels, right? 2 times pinnately compound. So here's that main stem, and then there's another little stem that's coming off the main stem, and then the littlest leaves are attached to that. So that's your 2 times pinnately compound. So if you go back here just to compare the 2, right? Oh, geez, come on. 1 times pinnately compound, 2 times pinnately compound. When you are pinnately compound, you can name the, so let's put it this way. I really want to like make you guys get this. This whole thing is a leaf. That whole thing is 1 leaf. This whole thing is 1 leaf. This is 1 single leaf. Because of that, you can't really call the parts on it a leaf because they're not. So what we call them is a pinna. So if you're 1 times pinnately compound, the little leaflets are pinna. If you're 2 times pinnately compound, you have pinna, which is the thing that's circled here, and then pinnule, which is each one of these little things. This is a pinnule, this is a pinnule, this is a pinnule, etc., etc., etc. And so you can have even more than 2 times pinnately compound. You can have like 10 times pinnately compound. And at that point, even botanists give up, because we're just like, it's too many things. So we have pinna still. So this one is 3 times here, but we can have even like 10 times. So it's 3. You have a pinna here, which is the first main division. And then on this pinna, you have these other little things. Here's 1, here's 1, here's 1, right? That we're calling a subpinna. But all you need to know is pinnule is the smallest one. So whatever is the smallest thing, here's the smallest, all of these here. The smallest thing is always called a pinnule. And the first division is always called the pinna. And then if you're 3 times pinnately compound, you'll have like subpinna. And then if you're 4 times, you'll have like subpinna. And then if you're 6 times, you'll have like subpinna. Even botanists give up. So don't worry about subpinna. Just know the first division is always called a pinna, and the last division is always called a pinnule. That's it. But you'll see sometimes like 3 times pinnately compound, and you're like, what do I call that thing? Don't even worry about it. Are we doing on time? Okay. This is a unique characteristic of the ferns, is the fiddlehead. So whenever a new leaf is growing on a fern, it always will show up as a fiddlehead, which is what you see here. And what's cool about fiddleheads is, look, here is a fiddlehead right here. Right? Here's one. What's cool about fiddleheads, here's one here. You can like curly Q, right? Little spiral. What's cool about fiddleheads is that only ferns have these. Only ferns. So if you ever see something and it has a fiddlehead on it. You could also call it a crozier. That's another term you might see for that. I like fiddlehead. But if you see a fiddlehead, you know 100% you're dealing with a fern, which is cool. It's nice to have such a identifiable characteristic for a whole group of a very large group of plants. And some fiddleheads are edible, which you can see here on the bottom. I don't know if I've ever eaten a fiddlehead. Now that I think about it, I should eat some to try them. I imagine they probably taste a little bit like an asparagus or something like that. That's what I would imagine. If anyone here has eaten fiddleheads, you can tell us what you think they taste like. So here's look, I took this photo here in the middle, growing out underneath this tree stump right here is a fern. The leaves are emerging. They're coming out as fiddleheads. And so I knew that was a fern emerging. Here's more. So in the spring, actually, it's coming up to be a good time of year to do this. In the spring, when things start to kind of melt and get ready to emerge, like all the leaves are starting to come out and stuff. If you go to kind of wet areas, you might see all these little curlicue things coming out of the ground. And that's fiddleheads. Those are fiddleheads of ferns. It's gonna be pretty cute. Here's some more. Okay, so fiddleheads, only ferns have them. Nothing else has them, only ferns. Of course, ferns are spore-shedding plants, and so we know they have to have sporangia. We know they have to have sporangia. Let's think about, just before we jump into the ferns, right? Let's have a refresh of where the sporangia were located on the other vascular plants we've talked about so far. So think about Selaginella, think about Lycopodium and Selaginella. Where on the plant were sporangia located on Lycopodium and Selaginella? Let's see, I'll pull up. Does anyone remember? So think about Lycopodium and Selaginella. Do they have seta? No, they don't have seta anymore. Yes, okay, got a bunch of answers in the chat box. On leaves of the Sporophylls, in Strobili, on Sporophyll's, on the leaves. Yes, good, okay. So yes, in Lycopodium and Selaginella, the sporangia were located on leaves. They were located on leaves, and we can call those leaves sporophylls. We could call those leaves sporophylls. Sometimes in Lycopodium, the sporophylls are arranged in a Strobilus. Sometimes the sporophylls aren't. Sometimes they're just on their own with the sporangia on top. And then in Selaginella, always arranged in a Strobilus, right? The leaves are always arranged in Strobilus. Okay, good. But the point is, in Lycopodium and Selaginella, right, the lycopods, the sporangia were located on leaves, right? Microphylls, and that in the case of those. Okay, so where were the sporangia located on the horsetails? Where are the sporangia located on the horsetails? Horsetails are weird. They're very weird. First of all, you know that in the horsetails, we just talked about it, their leaves are funky, right? Their leaves are like almost not leaves, right at the nodes. So probably not on leaves in the horsetails, because horsetails are funky. Okay, so yes, we have here Strobilus. I think we had a new term for it though. We did, because the Strobilus in the horsetails had these new structures that bore or carried sporangia, right? So sporophores, close. So they're bearing sporangia. Sporangiophores, yes, sporangiophores, exactly. So in the horsetails, we had structures that bear or carried, right, for sporangiophores. So we had that in the horsetails. Now we're in the ferns, and the sporangia are back on leaves. The sporangia are back on leaves. This time they're going to be on megaphyll leaves, right? Megaphyll leaves. And in the ferns, the sporangia are going to be located on the bottom side of the leaves. So we didn't really go into it in detail back then, but since we just went through it now, in the lycopods, right, Lycopodium and Selaginella, the sporangia were on the sporophylls, right? They were on leaves, but they were actually on the top of those leaves. If you were to do a cross-section through a Strobilus, you'd always see the sporangia on the top of the leaves. Now in the ferns, the sporangia are located on the bottom of the leaves. And one way this will be helpful to any of you who are going on to medical school or dental school, these terms ad and adaxial and abaxial. If you see, the way I remember it is ab with a B means the bottom. So for example, the sporangia are located on the bottom side of the leaves, abaxial. So when you see the B, it means you're on the bottom. It'll be helpful for you guys to who are going on to different scientific professions that use these terms. Okay. Not only, okay, so they're located on the bottom of the leaves, big deal, big whoop. But then here you got, this is the big whoop. In the ferns, the sporangia actually shoot the spores like little tiny catapults. Like, I don't know if you watched Game of Thrones or anything like that had catapults, but like, you know, catapults kind of like shoots it out. And the way it shoots things out like this is using these special cells that are sensitive to changes in humidity. That is called the annulus. This here is the annulus. When the annulus dries out, kind of pulls back. And then as it causes all this tension, and then it gets so much tension that it has to throw the spores out, throw them out. And then it recoils and it looks like how it did. Even though it's fun to watch me try to recreate this with my hands and things, it doesn't really do it justice. Here you go. So this is the bottom of leaves showing you, right, abaxial, the bottom side of leaves, showing you where the sporangia are located. And here we have another new term, which is Sorai. So in the ferns, usually the sporangia are arranged together. And when they are arranged together, we call the things they're arranged in clusters, which are called Sorai, the singular sorus. Sorai, not Sorai, Sorai. So like here, this hole here is one underside of a leaf. This is another bottom or underside of a leaf, and this is another bottom or underside of the leaf. Each of these, oh, blue is not a good thing to use on that. Each of these things, this is one sorus. This is one sorus. This is 3 Sorai. This is 4 Sorai. But each of these clusters is composed of sporangia, right? In this fern, in the middle, the clusters are like shaped like lines. So each of these lines is a sorus. All of these lines together are Sorai, but they're still composed of these sporangia. Lastly, over here, you see all these little orangey looking dots. Each of those is a sorus. Collectively, they're all Sorai, and they're all composed of sporangia. So the sporangian ferns are usually arranged in clusters called Sorai. Sometimes the Sorai, you have to remember, like humidity and temperatures and things being the right conditions are important for plants to know when they should release these spores. So in order to protect the sporangia and maintain a certain level of humidity, some species of ferns have a structure called an indusium. an indusium. All that is it's like I almost think of it as like an umbrella. Like that's kind of how I think about it. So if you look here, I'm gonna draw all right, so this here, we're looking at the bottom of a fern leaf. This here is one sorus, right? Here are 4 sori. And this has an indusium. Here's the indusium. And the indusium is almost like a little umbrella. Here it is. That covers the sporangia, kind of protects them, right? Here's another one. Kind of protects them from drying out. On the picture on the right, look, here's 5 sori, and each of these sori have almost looks like a hamburger bun type of indusium. So this little thing, it's like a little membrane flap that protects the sporangia. So the sporangia are underneath that. So that's a new term for us today, is indusium. Here's a better way. This might be clearer about why I think of it like an umbrella. So, oh, and just so you know, the plural of indusium is indusia, just so you know, it's not a typo on this one. So look here, this is the cross section through a fern leaf, right? Fern leaf. Here, this here is the leaf. This here is the leaf. Remember, the sporangia are going to be located on the bottom of the leaf. So here, all these, here's one sporangia. Here's another sporangia. Here's another sporangia. There's a whole bunch of them. Here's another one, etc. Right? These are all sporangia underneath here. We can call this cluster of sporangia a sorus, right? But this species also has an indusium, which, like I said, I think of as like a little umbrella, which helps protect them. So it's like an upside down umbrella because it's on the bottom of the leaf. But look, so this here is like an umbrella, right? Like if you're eating out at a restaurant and it's like super hot outside, you'll want to sit underneath something like this, right? So you don't dry out. And same thing goes here. So it's like almost like a little umbrella that protects the sporangia from drying out. Helps maintain humidity levels. Some species of fern have what's called a false indusium, in that they have on the edges of the leaf like folds over and protects the sporangia, but it's not really a whole separate membrane. It's just the edges of the leaf folding over to protect them. Oh yeah, oh wait, I gotta share this. So, hey, let's see if it doesn't work, then it doesn't work. Hold on, let's try. Oh, geez. Okay, hold on. Let's do this. So I'm going to try to play this video now, but if it doesn't play, just know that I put a bunch of videos on Blackboard, or I will today. And oh yeah, here we go. All right, hold on. No thanks. Let me share this real quick for you guys. Share. Stop share, and then I'll share this. Share. Make it big. And then we don't need to hear the person talking because they're just talking. But anyways, this is showing you, okay, be quiet. This is showing you underneath a fern's leaf. And this is not sped up. This is in real time. And what we're doing here is hopefully you guys can see the video. I think you can. We're zooming in to the bottom of these fern leaves. Now, the bottom of these fern leaves is where the sporangia are located. And in this species, the sporangia Sorai, here, I'm going to pause real quick. In this species, each of these things, so this is one sorus, right? This is whatever, 3, so these banana slug looking things, 3 Sorai, are the Sorai. And the Sorai are composed of Sporangia, right? And so what we're gonna see here in real time is as the conditions get drier, the indusium, oh, and this one is covered here. Look, I'll show you right here. Look, okay. So here, remember this whole banana slug thing is one sorus, right? Underneath this one has an indusium that looks like a big, almost like a big slug. So each sorus is covered by this slug-like looking indusium. And underneath here, you'll start to see the sporangia will start to poke out as the indusium starts to open up to let them poke out. And when they poke out, they're gonna start shooting their catapults. Okay, look, okay, there's one, this is one here. It's just about to shoot the catapult. That's sporangia. Again, not sped up, this is real time. Look, oh, it shot it. Okay, we're zooming in here. So look, each of those little things that's emerging are sporangia, and they're gonna shoot their spores. And this is in real time. This is in real time. This is not sped up. Starts to get good. Oh yeah, look, catapults. Look, boom, shooting the spores. Look, oh, catapults. Right? Wow, there's a nice shot of the annulus right there. Right? This is a nice shot here of an annulus. So here, look, this is the annulus of, here's another one, here's another one, here's another one of the different sporangia. Anyways, so there's a bunch of, or if there's not already on Blackboard, there's a bunch of videos that you could watch there that show you sporangia opening and shooting their catapults. Okay, I think we are going to finish one last thing, and we're actually going to get through this slideshow today, which I'm pretty impressed about, to be honest. All right. Yeah, so look for fern videos on Blackboard. Okay, so that was that. So far, we haven't really talked about the Gametophyte generation yet. You might remember way back in the beginning of this course, when we first learned about alternation of generations, we used the fern as the representative member to do alternation of generations. Remember heart-shaped green Gametophyte plant? Heart-shaped green thallus. So Gametophyte plant in the ferns, simple thallus, it is heart-shaped, right? It is heart-shaped. It is photosynthetic. Here's the heart, right? Heart-shaped. And it does have rhizoids, here they are down here, that attach it to whatever it's growing on. Remember, we said most fern sporophytes are homosporous, which means that most fern gametophytes are exosporic, right? Grow outside of the spore. And if that's the case, you usually have male and female sex organs located on one gametophyte plant. So that's the case in this example of ferns. Here you have the female sex organs, or the archegonia, are located near the notch of the heart. And the male sex organs, or the antheridia, are all spread out on the rest of the thallus. All right. We have already gone through the alternation of generations in the ferns way back in the several lectures ago, but let's just go through it again today using this diagram. This is a great diagram to have on hand when you guys are talking about ferns too. All of these like summary diagrams are really great. All right, so on the left, you have your sporophyte plant, right? Sporophyte plant. Sporophyte plant has things we talked about today, for example, horizontal stem or a rhizome. When the leaves, The megaphyll, remember they're the megaphylls, but when the leaves come out, they're always going to be in the shape of a fiddlehead. And in the case of this, look at this big, here's one leaf. And because there is so much stuff going on top of this one stem, we know it's a compound leaf, right? We can see it's a compound leaf. Anyways, we learned today that the sporangia are on the bottom or abaxial side of the leaves. So here, look, this is the bottom of one of those leaves. And the sporangia are arranged in clusters called Sorai, which are these little dots that you can see on the bottom of the leaf. Notice this, So if we do a cross section through this leaf, here we can see one sorus, right? One sorus. Notice there's no indusium in this species, so the sporangia are just exposed. See, here's another sorus. There's no little umbrella, right? No little umbrella. Not every species has indusium. Okay, here's one sporangium. Here's one sporangium. It's on the bottom of the leaf. Eventually, it's going to, via humidity changes, shoot its spores like a catapult. Shoot its spores like a catapult. And then once that happens, right, spores are going to be released. The spores are going to grow exosporically or outside of the spore wall, right? Sorry, the gametophytes are going to grow exosporically or outside of the spore wall. So like this darker thing here is a spore wall. Darker thing here is a spore wall. Thing emerging from it is the gametophyte plant. All right, now our gametophyte plant, heart-shaped gametophyte plant is all grown up, and it has on it male and female sex organs. Sperm from the antheridia are going to fertilize the egg in the archegonium. Here you go. Here you go. Here's an antheridium that's open. Sperm leave and swim out, fertilize the egg, and then your result is a 2N zygote via fertilization. Sorry. 2N zygote is going to grow, and ultimately it'll first be attached to the sporophyte, remember, the egg was in the gametophyte plant, right? And so when the sporophyte plant first starts to grow, it's still attached to the gametophyte plant because that's where the egg was. But even though it's briefly attached to the gametophyte plant, the sporophyte plant is not dependent on it, because as soon as the sporophyte plant makes a leaf and makes one root, then it can get its own food and water, and so it doesn't need the gametophyte plant. And then sporophyte plant will grow into mature sporophyte plant. And then we're back at the beginning. So pretty easy. This one's a little bit more straightforward. That's why we use this as an example in the class. Okay, we got through more than I was expecting to get through today, so that's good because it'll put us in good shape, kind of lessen the load of material coming as we get closer to the exam, which will give us more time to review and kind of take a chill pill and relax a little bit. So next time, we'll actually talk about some specific fern species, and we'll also have a general discussion about like, how do you study next class? And that's it. Just a reminder, if you came late, if you missed assignment 3 this morning, send me an email so I can reopen the assignment for you. And I will take any questions that you guys have. Otherwise, have a great rest of your day. Yeah, Mohammed?
Yes, so I did assignment 2, and what my grade when you review it, it got down. Like it was in Blackboard 16 something, and when you review it, it came down. I don't know why, and I didn't review it even.
Right, so I can relook at your assignment, but so what happens is when you submit an assignment to Blackboard, Blackboard tries to grade it. Blackboard tries to grade it, but sometimes Blackboard doesn't get it correct all the way, and so I manually open each assignment after it's submitted and grade it and correct any errors that Blackboard made.
Oh, okay. So sometimes even Blackboard do mistakes or something for grading.
Yeah. So for example, sometimes there's more than one correct answer. So like sometimes there's like two answers that are both correct, and for some reason, Blackboard, if you select, I still haven't figured out, like if you select one of those, Blackboard will be like, yeah, you got it all right. But then you might have also selected others that are wrong in that same question. Do you understand what I'm saying? So like,
yeah, I understand, but
yeah.
I didn't even have I didn't look at my grade, the questions to see which one I got wrong.
Okay, cool. I think you should be able to see them now.
Okay.
Yeah, because I opened them on Tuesday, so I think you should be able to see all, not only what you responded, but what all the other options should be.
Okay.
Yeah. Okay. Yeah, and if you look at it and you still, I should say this for everyone. If you ever think I made a mistake in grading, like, so if you pull up your assignment and you think like, oh, I don't know why I got this wrong, feel free to email me so I can double check that I didn't make an error, and then add back points if need be. So that's also totally fine.
Okay, thank you. Happy weekend.
You too. Thanks. Yeah, you guys have a good lab tomorrow, okay?
Professor.
Yeah.
What exactly is a sporangiophore in a horsetail?
What does a sporangiophore do?
Or what is the sporangiophore? Like I see that what were we just talking about?
Here, I'm gonna pull up that, yeah, here we go.
Like I see ferns, they have Sorai, but I don't know what the sporangiophore looks like on a horsetail. Like I know lycopods, they have a Strobilus. Well, not
Yep.
even. So Stylogenella have strobili.
Yeah.
But yeah, I don't know what a sporangiophore looks like on a horsetail.
All right. Let me pull this up. Hold on. Yeah, it's kind of like horsetails. Why you gotta be difficult? Like, why can't you just, why does it have to be like a whole new thing with the sporangiophore? It's kind of silly, but okay, hopefully you could see this. Let me make it big. Okay. Can you see the kind of alternation of generations of the horsetail here?
Yes.
Okay, great. So let's just look here real quick. Annotate. I'll go with red. So over here, I should do this. This whole thing here is a horsetail sporophyte plant, this whole thing. And at the tip of this stem, because remember, this species has some stems that just do photosynthesis, and then other stems that have the sporangia on them.
Right.
On the tip of this stem, we have a term we're used to, which is a Strobilus, right? And what I will say is, remember in the lycopods, so here, I'm gonna see if I could do this. In the middle here, I'm gonna draw something, okay? So in the lycopods, I'll do, oh, I wonder, can I do text box? Ooh, I can't, hold on. Okay, so in the lycopods, which I'm going to put here for a second, we're just doing a breakout right here. They had a Strobilus as well, but in the lycopods, the Strobilus was a stalk with leaves on it, right? A stalk with leaves on it that we called a Strobilus, and each leaf is a sporophyll, right? Right? Okay. But in the horsetails, as a comparison, where's my text box? Here, horsetails. In the horsetails, the Strobilus now is a little more complicated in that it has this new structure. So in the horsetails, you have a stalk. And on that stalk, you have this thing called a sporangiophore. I'm going to draw it like this. And on the sporangiophores, you have the sporangia underneath, like 1, 2, 3, 4, whatever, 20 on each one. And I'll show you what they look like next, but I'm just making this distinction. Whereas on the lycopods, since the Strobilus in the lycopods is just one made of leaves, each leaf only had one sporangia, right?
Okay.
Right?
Yes, thank you.
Okay, so but then look. So here, let's go next. Here's that top of the horsetail Strobilus. This is the top of that horsetail Strobilus right here. And here's a cross section through it. And this is what I was just trying to draw, although not very well. Here's the stalk of the horsetail Strobilus, and then each of these mushroom looking things. So here with this, here's another one here, kind of like mushroom looking things, are those sporangiophores, right? Here's a whole one right here. And here's if we pulled off the sporangiophore, this is what it looked like. And hanging off of the sporangiophore is all the sporangia. So each of these almost looks like bananas coming down are the sporangia, hanging off the sporangiophore. In terms of what does it look like in real life, if we go here, this is what they look like in real life. So they don't look like leaves at all. They almost, to me, look like little mushrooms. So here is that same Strobilus that we saw on the previous slide of the horsetails. And each of these things on it is a sporangiophore. Here's a close-up of a sporangiophore. Almost looks like a little mushroom. And hanging off of it, these lighter things, those are the sporangia.
Thank you so much. And those are homosporous, right?
They are homosporous. Yep. And the thing, the reason why we go over this thing is because it's more efficient. If you have a sporangiophore, each sporangiophore can have on it sometimes like 20 sporangia. Whereas in the lycopods, when you had a Strobilus that was just made up of sporophylls, each sporophyll can only have one sporangia on it. So it's like, if you have, let's say on a lycopod, you have a Strobilus that has 10 sporophylls on it. That means you can only have 10 sporangia on it. But let's say you're a horsetail and you have a Strobilus that has 10 sporangiophores on it. And each sporangiophore has 10 sporangia on it. Now you have 100 sporangia as versus 10. And that's why. Yeah, no problem.
Have a good day. Thank you so much.
You too. Have a good one. Have a good weekend. Have a good lab.
Hey, Professor, I had a quick question.
Yep.
So I was growing this basil plant and it died. Is like basil, like, is it kind of like a perennial? Like, is it going to come back next season or no?
No, hun. No, not if it's dead. It was growing inside.
Yeah, it was growing inside, and then all of a sudden it just started dying, but like I didn't change anything. I tried putting it closer to a window so I could get more sunlight,
Mhm.
and I would just water it like usual, but it just kept, dying.
Yeah. How long did you have it for, Andrew?
I think I got it like in the summer, towards the end of the summer.
Okay. Yeah, I'm sorry to hear that. It could be just like basil likes to have a ton of sunlight. Like they like to just be out in the sun like for 6 to 8 hours a day. I've also, for the record, I've also had several basil plants over the last 6 months that, I mean, I pick at them to make food, but also they've also passed away. So, yeah, I think it's just they don't like being inside without a lot of really hot sun for a very long period of time. So like they'll hang out for a few months, 6 months, maybe a little bit more, but unless they're getting like 6 or 8 hours of direct sun a day, eventually they'll just be like, nah, done.
Okay.
Yeah, a lot of herbs are like that. They just like, they want so much sun. One thing you could do is you can use clamp lights, or you could, so what I use sometimes is I go to the hardware store and I get a clamp light.
Hmm.
And then they also at the hardware store sell different like spectrum bulbs, and some of them are like, oh, this is the daylight,
Yeah.
daylight bulb. Not a fancy plant growing light bulb, but just a daylight bulb. And using that with the clamp light, you're looking at about maybe $10 total.
Uh-huh.
And you can use that to shine on plants that want more light than you can give them with the sun.
Oh, okay.
Yeah.
All right. Thank you.
All right. Take care, guys. Have a good
Bye.
weekend. Bye.